Howdy, folks. Welcome to the second recording for week 4 of our Bio 5 class. So there should be a recording posted for chapter 19. This is the recording for chapter 20. Information from both videos is going to be included on our exam, which is going to be due 1 week from tomorrow. I'm recording this on Saturday. It should all be open by Monday. You can do everything in your own time remotely. Uh, Honestly, I don't know if I need to do this preamble. You just watched the other video. So I think we're just going to go and get right into it. Screen two, and then pull up the presentation, and here we go. Alrighty, so this is chapter 20, which is our chapter on genetics and human inheritance. We're starting to get closer to my specialties now. Cell biology's never been really my chuff. So I think that this will go and be a little bit more of a smooth video than we've been getting. So, framework wise, we want to go and hit a couple different points. Firstly, we want to talk about the principle of inheritance, the break in chromosomes, and detecting genetic disorders and how we go about that. So, a little review from our previous video. Genetic information we know is carried on chromosomes. You get one copy from your mother, one copy from your father, and equal number in the eggs and sperms. And that we have 23 pairs of homologous chromosomes, one set from each parent, with one of those being sex cells and 22 of those being somatic non-sex cells. So genes are segments of DNA that code for a specific protein. Proteins play a structural or functional role in the cell. Traits are different characteristics that are controlled by gene-directed proteins. This can be controlled by a single protein or many, many ones. Alleles are different forms of a gene that produce different versions of that trait. So, for instance, there's a gene for freckles. One form of that gene, one allele of that gene, causes freckles to form, and one form, a different allele, causes freckles to not form. If we look at a chromosome here, a gene is always going to occur at the same spot on a pair of sister chromatids, and a gene is always going to be a region. Variations within that gene are going to be our different alleles. Because I get one copy of DNA from my mom and one from my dad, I am getting both copies of this. I could have different copies of the allele from each parent. Now, if those copies are the same, they are what we call a homozygous pairing. So homo is from the Latin for same, zygo means to join, homozygous, same joining. So that's where you have two copies of the same allele. Heterozygous, from the Latin hetero for different, is where an individual has different copies of the same allele. So this would be like I have one copy of the freckle gene and I have one copy of the no freckle gene. They both are the same gene. I just have two chromosomes, so I can have one of each. Now, if one of those genes is capable of overriding the expression of the other gene, let's say if you have the freckle gene, you are 100% getting freckles no matter what. We call that a dominant gene. The gene that can be overwhelmed by other alleles is called the recessive gene. So the no freckle gene, because if I have both the freckle gene and the no freckle gene, I will only present with freckles. The no freckle gene is not being expressed, so the no freckle gene is considered recessive in this relationship. Genotype is the genetic composition of an individual. It is their particular alleles for every single gene in their body. Phenotype is the expression of the genotype. These are the observable phenotypic traits of an individual. So, with the freckled person, if I have the freckle gene and the non-freckle gene represented by a capital F and a lowercase f, respectively, then I'm only going to have the freckled phenotype because that's the only one that's presenting physically and is observable. So here, this person has freckles. Freckles are dominant, so this is either FF or FF. In other words, dominant F or heterozygous F. This is a homozygous recessive individual for the freckle gene, so their skin has no markings. Widow's peak is also a dominant trait. So it could be either the homozygous dominant or the heterozygous pairing, while only the homozygous recessive can result in a straight hairline. Unattached earlobes, attached earlobes, and tongue rolling, all examples of the genes that have a simple dominant or recessive relationship. These genes, this, mind you, might not determine the expression of these genes in the population. If any of these genes are deleterious and hurt the reproductive ability of the individual, let's say I am an earlobe fetishist. I only am attracted to people that have attached earlobes. Well, there's now selective pressure for attached earlobe people to get more babies. There is unattached earlobe people are not part of that trend. So you might see a real world example of the recessive attached earlobe gene occurring more often than unattached earlobes. It just has to do with historical pressures, anthropological pressures, and small stuff. You know, if it happens to make you look good, it's probably going to help you reproduce. So this just describes this type of relationship. Here you can see the genotypes for the allele, one from mom, one from dad, one from mom, one from dad, and one from mom and one from dad. The description of these allele and the phenotypes that are observed. And again, even though these have different genotypes, they present with the same phenotypes over here. Gamete formation follows what is known as the law of segregation, which is that during gamete formation, the two alleles for each gamete, for each gene, separate. They do not go to the same final destination. The law of independent assortment basically just states that this is happening randomly and arbitrarily. Like if two genes are on separate chromosomes, they do not influence each other's division. If they are on the same chromosome, though, they most likely will be inherited alongside each other. So here is the freckle gene and the no freckle gene. And this is basically just labeling each chromosome by what genotype it has. So this one is dominant for freckles, as is this one. This one is recessive for freckles, as is this one. So we duplicate, and then we undergo division to create our polar bodies, and we end up with kind of a random assortment. We end up with a random assortment of the genes that were created. Now, we don't know which one of these sperms is going to go to which one of the is going to go and make it to this egg. And this egg also had an equal chance of forming as any of these other polar bodies did into turning into an egg. So the sperm will go and penetrate, fertilize, and now we have gotten many stages of randomness introduced into our reproductive process. This randomness encourages and accelerates the process of evolution and increases genetic variety inside of a population. So simple genes, like the ones that we've talked about so far, are usually controlled by a single point. And they often use a dominant recessive relationship. This is not how all genes work. This is not even how most genes work, but this is the best way to go and get into the room and start talking about genetics. So Mendel was a scientist, one of the earlier, who was an Austrian monk in the 1600s. He aggressively selectively bred pea plants and created a statistical projection tool in order to understand natural rates by which genes would be inherited between generations. We call this tool a Punnett square. It's a form of matrices, and it can help predict the genetic makeup of an offspring by giving you a probability estimation for an offspring's traits. This is more of a farming tool than anything else. Like, that is its best use. It's really useful. If you breed pigs, if you breed chickens, if you breed cows, if you breed anything, if you breed different plants, you're probably at some point going to need to go and pull out a Punnett square to make sure that you don't waste a lot of money. So Punnett squares can be divided into different components. It's easier to explain these keywords with the actual diagram itself. So this top row is going to go and have the genetics from one parent. And because this parent has two alleles for this gene, one is going to be assigned to each of these squares. So top row, male genes, In this case, the gender doesn't really matter. You just have to make sure that it's only one parent's genes, evenly divided. The female in this case is a homozygous dominant, so her genes are evenly divided against the side column. Like this is gene one, or sorry, allele one, allele two, allele one, allele two. And then from there, we can simulate breeding by just recombining the genes. Okay, well, we know she's going to go and put out dominant F eggs, and he's going to put out recessive F sperm. So when they link together, they're going to create they have 100% chance of producing offspring that are heterozygous instead of homozygous for that trait. Now, we start to get weird ratios when you start to change the alleles. So in a monohybrid cross, both parents are heterozygous for one trait of interest. So they'd have a genotypic ratio, and that's just one big F, one little f, one big F, one little f. That leads to a ratio of one homozygous dominant, two heterozygous, and one homozygous recessive individual. So Mendel did this experiment with pea plants that had a dominant purple color and a recessive white color, and he was able to observe these ratios as he was doing his experimentation. And again, these are not guarantees, these are probability projections. There's a 50% chance that you'll have a heterozygous child and a 25% chance you'll have a homozygous child. I don't know about you, but probability's never been my friend. I've never won the lottery. So you are most likely, so you could end up getting like 10 homozygous recessive children. It's just less likely. So we refer to two heterozygous individuals being crossed as a dihybrid cross. Oh, I'm sorry. So monohybrid cross, both parents are heterozygous for one trait of interest. In a dihybrid cross, we actually keep track of two separate genes at once. So if both parents are heterozygous for both genes, they actually end up with a phenotypic ratio of 9:3:3:1. So I'm going to really quickly show you an image of what this might look like. So dihybrid heterozygous cross peas. All right, here we are. So this is the Punnett square that we end up with for this particular scenario. So we start with two heterozygous parents, and each one of these squares is just a possible combination of the two genes that each parent could go and give. Each one of them is going to have two copies of R and two copies of Y, but each of their daughter cells they're producing can only have one copy of Y or one copy of R. They can't have both. So these are all the possible combinations that could be produced through random assortment. Each one of these squares has an equal chance of occurring as any one of the other squares. Anytime we get a higher number, like let's say these guys, they show up 4 times. So that means it has a 1 out of 4 chance of occurring, a 25% chance of occurring. This one only occurs once, so it only has a 1 out of 16 chance of occurring. Yeah, so here's like so here's dihybrid cross as well. And again, you can see how they came about through that 100% allelic reshuffling we were talking about earlier, and then what happens when we cross that with itself. And we end up with this specific phenotypic ratio, 9:3:3:1, where this is homozygous recessive, homozygous dominant, and then 2 different types of heterozygotes. Alrighty, cool. So let's get back to the presentation. All right. So both parents are heterozygous for 2 traits of interest. We got the appropriate phenotypic ratio. This is a pea plant thing. Here's a visual example. So these individuals both carry the freckle gene. Or sorry. This is one freckled female mating with a non-freckled male. When we go and look at here is a example of how this might manifest between 2 heterozygous individuals, 2 freckled individuals. end up with the exact same ratio that we ended up with over here because it's a mathematical function. It doesn't matter what these genes are. They're just but this is an analogy. So let's have a look at this video and that can explain things in a bit more detail. Didn't realize there was a video. That's why I was confused.
During the 19th century, the Austrian monk Gregor Mendel performed plans and worked out much of what we know today about the laws of heredity. We will use his ideas to consider the inheritance of a single trait, freckles. The inheritance of freckles is determined by a dominant allele. Suppose a freckled female who was homozygous dominant, big F, mated with a homozygous recessive male with no freckles, little f. How likely is it that their child will have freckles? A Punnett square helps determine the probable outcome of the genetic cross. Both parents are homozygous, so each can produce only one type of gamete as far as freckles are concerned. The female produces gametes with the dominant allele, big F, and the male produces gametes with the recessive allele, little f. Each square in the resulting table is filled in by combining the alleles from the sperm and eggs. In this example, all the children would be heterozygous for the trait, big F, little f, and therefore would have freckles.
Now,
Now let's consider why it's possible for parents who are both heterozygous for the freckle trait to have a child without freckles. The dominant F and recessive F alleles segregate during meiosis, so half the gametes bear the dominant F allele and half bear the recessive F allele. Filling the boxes, we see that the probable genotypes of the children would be in a genotypic ratio of 1 homozygous dominant, big F, to 2 heterozygous, big F, little f, to 1 homozygous recessive, little f. Phenotypically, the children would be 3 freckled to 1 not freckled. A child from these parents would have a 25% chance of not having freckles. Suppose 2 people who are heterozygous for widow's peak and freckles mate.
oh, whoops, sorry. This is the example that's going to go. That is the dihybrid cross. So let me just get to the beginning of that.
Dominant F allele and half bear the recessive F allele. Filling the boxes, we see that the probable genotypes of the children would be in a genotypic ratio of 1 homozygous dominant, big F, to 2 heterozygous, big F, little f, to 1 homozygous recessive, little f. Phenotypically, the children would be 3 freckled to 1 not freckled. A child from these parents would have...
All right, did we... I feel like we skipped something. Oh, no, we didn't. Okay, we were right here. Sorry about that.
Freckles. Suppose 2 people who are heterozygous for widow's peak and freckles mate. Freckles and a widow's peak are both controlled by dominant alleles, so these heterozygous individuals show both traits. Would it be possible for this couple to have a child that had neither freckles nor a widow's peak? We can use a Punnett square to analyze a 2-trait cross, as long as the genes for the 2 traits are on different chromosomes. Keep in mind that during gamete...
It's important to note that if a gene is on the same chromosome as another gene, they are most likely going to be inherited together during independent assortment, unless those genes were crossed over.
Segregate independently of one another. The gamete. Dominant F, dominant W. Dominant F, recessive W. Recessive F, dominant W. And recessive F, recessive W. Each type of gamete has an equal chance of joining with any other type in fertilization. Numbers of phenotypes possible for the next generation are 9 freckled with widow's peak, to 3 freckled with straight hairline, to 3 no freckles with widow's peak, to 1 no freckles with straight hairline. The expected phenotypic ratio is 9 to 3 to 3 to 1.
So phenotypic ratios are the way that we measure these physiological traits. Here you can just see, so it is literally just a ratio of the frequency of occurrence. Reduce this down to the minimum in general. Like if you can get one, like if it's a 2 to 4 to 4 to 2, you should write down 1 to 2 to 2 to 1. Just simplify. Here's an example of how that's processing in the terms of meiosis and mitosis. And this is just a standard Punnett square for two heterozygous individuals. And this is what's happening on the cellular level during this to kind of help you understand how the probability projection comes to be. And here's a visualization of the dihybrid cross, keeping track of two independently assorted genes together and the phenotypic ratios that we end up with from that. Now, pedigrees. So a pedigree is basically genetic lineage for certain genes that are tracked throughout a family's throughout a family. So super common for tracking royal lines, especially once we realize that like, oh crap, inbreeding is making all the royals have water for blood and they're all bleeding out. So this kind of helps avoid some of the more deleterious effects of things like that. It's still used by a lot of island communities just because they have a smaller breeding population and it's more important to double check to make sure you're not marrying your cousin twice removed. Like I know that the state of Hawaii has a couple registers like this. Iceland has a register like this. A lot of the Pacific Islands, Madagascar, pretty much just stuff to make sure that people aren't that their gene pool doesn't become a gene puddle. So here is a this is tracking for so this is the pedigree for a dominant trait, and this is the pedigree of a recessive trait. Individuals with the visible phenotype are highlighted in blue. Squares are boys, circles are girls. So you can see how a recessive trait can actually stay hidden for a very long time in a lineage. As long as nobody is homozygous for it, it's not going to physically manifest. But a dominant trait is going to occur in every single generation. So this is actually one of the indicators that we use to go and to figure out if a trait is dominant or not. If it keeps showing up over and over again, it's probably dominant. Genetic disorders are often caused by recessive alleles because that is the only way that a deleterious mutation can be passed on. If it affects your ability to live and it's a dominant trait, it's going to be selected against very quickly. If it can stay hidden and it might have a secondary useful trait when it is recessive, like a lot of the times, a genetic disorder is like if you only have one bad copy of the gene, it can actually help you. That's sickle cell does that. If you are heterozygous for the sickle cell gene, you're immune to malaria, but you don't have any of the negative symptoms. If you're homozygous for it, you have like chronic fatigue and a lot of circulatory issues, and you might end up dying. Dominant allele produces a functional protein, while the recessive allele does not. Right. Sorry, I was hitting the wrong button there. I think I confused myself. There we are. All right. So dominant alleles produce a functional protein. Recessive alleles do not. Uh, so like a good example, this would be albinism. So if you do not have the ability to produce melanin, uh, that's a that's a homozygous disease and homozygous disorder because the dominant allele results in pigmentation, which means that albinism can skip generations. If an albino person reproduces, then their kids have a chance of having albinism, but only if that albino person is reproducing with someone who carries the albino gene. Yeah, when it's homozygous recessive, you end up with an albino. We know this very well because we bred a lot of albinism is one of the easier traits to breed into like a lab mouse. So we've studied this pretty extensively. Complete dominance is when a heterozygous trait completely overpowers the recessive trait. Like we only are seeing freckles. We don't see a new physical being. It's either freckles or no freckles for the particular capital F, lowercase f freckle gene. Codominance, however, is where alleles from both parents are present. So let's say I have a big B allele, which is for blonde hair, and a little B allele, which is for green hair. If I have codominance, I'm going to produce hair that is blonde as well as hair that is green, and both are going to be fully expressed. By contrast, in incomplete dominance, I end up with something in between. So instead of my hair being blonde and green, and each strand being blonde or green, it's going to be kind of like a muddy color that's an averaging of the both. So incomplete dominance, both genes are partially expressed and they kind of fade out. Occasionally, we'll have a gene that does many, many things. These genes are known as pleiotropes. So sickle cell, we've already talked about sickle cell. It basically acts a lot like diabetes because it's making jagged shards that result in reduced circulation and shredding of little blood vessels. Same way the sugar crystals in your blood do when you're a diabetic. Here's the genetics for sickle cell. So there's the functional gene and the non-functional gene. The non-functional gene codes for sickle cell. The functional gene codes for functional blood cells. If you're a heterozygote, you're immune to malaria. If you are a homozygote for sickle cell, you're anemic and sick, and you might lose your feet. And if you're homozygous for the normal one, congrats, you just match every other person outside of a malaria region. So here you can see how that functions through. The most important issues here are it just catches on stuff. Every single one of these is a clot. All these are symptoms of clotting. So occasionally multiple alleles, sometimes up to 15, of different genes exist in a population. So a good example of this is for blood typing. We have type A, type B, and type O, and those are three separate alleles. Blood typing is kind of unique and interesting because your blood type's actually determined by two unassociated genes. Because you have a gene that controls your positive and negative, which is a nice simple dominance relationship, and then you have the multiple alleles with the three types. And it's interesting because there's co-dominance between type A and type B, but type O is recessive to both. So if you have someone with AO blood, they present as type A. If you have someone with AB blood, they present as type AB. And the only way to have someone who presents with type O is for them to be homozygous for type O and type O. Interestingly enough, my sister is type O negative and I'm type O positive. O negative is the rarest blood type because the positive gene is a dominant over the recessive gene. Pretty cool, if I do say so myself. Alrighty. So the markers that they're talking about are these polysaccharides that are on the surface of blood, and these are chemical signals. If you do not have the right kind of blood and you're given a blood transfusion, you'll end up having an immune reaction to it because your body will treat it as a foreign agent and it'll end up killing you. And again, it's important to emphasize A and B are codominant, O is recessive to A and B. So here are the different genotypes that can be possible. Here are some of the different genotypes that are possible. And here are all the phenotypes that correspond to those different genotypes. So here's double I, or double lowercase I, which is the genotype for type O blood. AB, type AB. All Bs with a recessive O, it's going to present as B. And all As with a recessive O, it's going to present as A. So occasionally we end up with what's known as polygenetic inheritance, which are variations within a trait that are independent of environmental influences. They involve two or more genes, and they can occur on different chromosomes. Very complicated genes that have kind of a spectrum of expression, like skin color or eye color, tend to go and hinge on this. When I was a kid, I remember there was a rumor that went around that if your parents had brown eyes and you ended up having blue eyes, it meant you were adopted. Well, we found out that there's like 30 genes that influence like blue eyedness alongside like 200 other genes that influence other eye colors. So, like, when people were saying stuff like that, they were just picking on a child, which, while funny, is wrong. You shouldn't do that. Anyway, so some of the more common examples of polygenetic inheritance are height, skin color, and eye color. There's a continuous spectrum from the smallest to the largest, from the darkest to the palest, and then from the multitude of variation of eye colors. So this is just because there is such an extreme volume of traits associated with expression of that gene. Like, here are some of the alleles that we assume influence height. So this is one gene, two gene. These are all different combinations, which leads to a lot of kind of crazy variants. You'll notice when multiple genes are involved, because these are subject to independent assortment, we start to see some kind of standard statistical trends emerge. Here, you see a bell pattern where the tallest are going to be in the middle, and then we're going to have less in the extremes of tall and short, because those are less useful than being average height. So me, I'm 6'2". I have heart trouble. I have joint issues. I have to be super careful about my weight because it's super easy for me to get to a point where my heart is going to start struggling. I get 10 pounds overweight, I have high blood pressure. That is because I'm disproportionately tall. By the same token, someone's 4 feet tall. Well, you can't really do a lot. You kind of lose a lot of options for activities, including, especially in a primordial society. Like it would have been selected against if it would have gotten in the way. All of this is just to say things that make you unhealthy and things that this curve is evidence for evolution. There are selective pressures that are pushing us towards the central line. It's fascinating. So genes that are on the same chromosome are called linked genes. They tend to be inherited together because they tend not to independently assort, but they can be unlinked during crossing over. So it's not like a guaranteed thing. And even then, it's not like a set rule. It's just where did it split? This is part of the reason why we're having so much trouble understanding what every gene does. It is a math problem that needs to be solved through computation. So genes on the same sex chromosome are linked. Sex-linked genes tend to cross over less, so they're some of the more well studied. They also are easier to mark because we have the X and the Y that are so visually different and have such a defined relationship that there's a lot more study around this. Y-linked traits are incredibly rare just because the Y chromosome mostly is modifying traits that are presented from the X chromosome. Like the Y chromosome says I need to grow a beard, but that's just telling the hair follicles coded by different chromosomes to speed up production of a certain hormone. Uh, X-linked disease, so sex-linked diseases tend to be X-linked. I can't actually think of a single instance of a Y-linked. I just am, as a biologist, you should never speak in definitives. Now, X-linked genes more common because there's more genes on the X chromosomes. Males only have one X chromosome. Females have two X chromosomes, which means that if a male inherits a recessive sex-linked disease, they are going to express the phenotypic traits. If a female inherits one good copy from an X-linked, if it's a recessive sex-linked disease, then she's not going to present with it and she'll be a carrier for it. So a couple diseases that tend to follow this pattern are red-green color blindness, hemophilia, and muscular dystrophy. They tend to be X-linked. They tend to be passed on through the female lines because if the male had this trait, it would be manifesting. It would be interfering with their breeding fitness. However, some of these don't really affect individuals very much. Uh, my grandfather was Amish. He was red-green colorblind. You don't really need to know a lot of colors in order to go and be a decent farmer. So he did just fine. And here's an example of how this might work in a little bit more detail in a different model.
This produces a particular pattern of inheritance because females have two copies and males only one copy of the X chromosome. X-linked characteristics are therefore expressed more often in males than in females, and as a consequence, X-linked diseases are expressed in more male than female victims. Red-green color blindness is an example of an X-linked recessive disorder because color blindness is not expressed if a functional allele is present. On the Punnett square, we can denote the X chromosome with the normal, functional allele with a black X, and the X chromosome with the defective allele with a red X. The possible progeny are XX, both normal alleles, red X, black X, carriers with one normal allele, black X, Y, normal males, and red XY, colorblind males. Note that the genotype must be either red X, female, or red XY to express colorblindness. A female of genotype red X, black X has normal vision and is a carrier for the disorder. Although she will not express the recessive trait, she can pass the defective X allele to her offspring. As this example illustrates, the chromosomal makeup of males makes them more susceptible to X-linked recessive disorders than females. For a daughter to express colorblindness, she must inherit two recessive alleles, genotype red X, but a son who inherits a single recessive allele will have the colorblind genotype red XY. As a consequence, colorblindness is much more common in males than in females.
Yeah, so it's just a fascinating trick of probability in mathematics. Like at the end of the day, these are all probability projections. Anytime you see a Punnett square, that should be your first thought. And it just comes down to one thing. You are only have one possible X chromosome from a father. You have two possible X chromosomes from a mother. If you're a male, and you get a bad copy, you're screwed. Sex influence genes basically include genes that might not be on the sex chromosomes, but are influenced by sex hormones. So your hair cells, like your hair follicles, if you have too much testosterone, the ones on your head will reduce production, while the ones on your face and neck and body will increase production. Your follicle cells are not sex cells. They are not gametes, but they are influenced by a sex hormone. This is the foundation of sexual dimorphism, the phenomenon by which a male and a female of a species have different physical traits. Humans and primates in general have pretty exaggerated sexual dimorphism due to historical and anthropological reasons. But we have noticed that of the primates, we have the smallest degree of difference in size, which implies a certain amount of anthropological equality in the way that we distributed tasks in the past. Like it is more exaggerated in species where the gender is more have different life cycles. So like an elephant seal is 8 times the size of a female because they're going to different like oceans. One's going to the Antarctic, the other's going to hang out in Hawaii, as well as, different selection patterns for breeding. The point is, there's a lot of examples of these in nature. It's interesting to go and learn about them. So male pattern baldness is dependent on the presence of testosterone. If you have to go and have the allele for baldness, which is a sex-linked gene inherited through the maternal line. So if your maternal grandfather is bald, you have a risk of being bald. If your maternal grandfather has most of his hair, you are not going to get genetic baldness. If you start abusing testosterone, it can bypass, or you get a disorder that causes you to produce too much testosterone. It can bypass this stage and just induce hair loss, whether the allele is present or not. It can develop in women if it is a homozygous trait, but they would have had to have inherited that trait from a bald man and a woman that either was bald herself or carried the baldness gene. So it's much rarer, and it requires a bald man to be involved in order to make a bald woman. Occasionally, chromosomes can be damaged. This can be caused by viruses, radiation, just issues with the division of a cell, like, and of course, chemicals as well. We call them carcinogenics. And this results in the change in the structure and function of the chromosome in a form that we call a mutation. So, in mutation, we got a couple different types. We have a deletion, which is where a chunk of a chromosome is removed. The most common that occurs is where a tip breaks off a chromosome 5. This occurs surprisingly frequently, and it leads to a neurological disorder known as Cri-du-Chat syndrome, or crying baby or cry of the cat, because it leads to babies that have brain and throat deformities, and this causes them to sound like a cat when they cry. They kind of meow at you. They are severely mentally inhibited. They will not be able to function at a normal level. It's a pretty rough disease to deal with. So duplication occurs when a chunk of a chromosome is replicated and then attached to a different chunk of a chromosome. The effect depends on the size and the position. There are a lot of different versions of this. Occasionally, one of the more common ones is Fragile X syndrome, which is duplication of a region on the X chromosome. It leads to some interesting physical traits, like the teeth come in differently. The ears tend to go and be flared. Usually, mental retardation is not a component of it, but it has occurred, and it might require some therapeutic assistance in order to help rehabilitate the individual. But yeah, it's a fascinating disease. And also, man, that guy has really blue eyes. Good on him. All right. So, if an individual's older than 35, there is a probable chance that prenatal genetic testing is going to go and be is going to be necessary, because once you hit about 35, you start to have increased risks of non-disjunction. Now, I've heard people, hell, I've known people who act like their life is over when they hit like 30 or 35 because they're like, I don't have time to have kids. Well, the reason we do this genetic testing is because we have very effective interventions that can lead to that can lead to a safe and healthy baby, as long as we can apply a medical treatment in time. So it's good to go and get the genetic testing in order to go and try to catch non-disjunction, because it can be corrected and it's reasonably easy to correct. And that doesn't mean your life's over when you're 45. If you really want to have a kid and you can afford it and you think you're going to go and survive to adulthood, doesn't really matter how old you've gotten at this point. We can probably make it work as long as you're younger than 50. So aminocentis is the main way that we go and test the genetics of the fetus. So we just go and take a little bit of fluid from the amniotic sac, don't actually touch the baby. And then we culture cells from the fetus and make a karyotype from their chromosomes. This in turn lets us go and eliminate, and sometimes we'll even sequence the DNA from the baby to go and look for certain things. But in general, we can catch a lot of stuff early enough that we then can go and that we then can fix it. Now, there's another type of sampling called chorionic villi sampling. So this is a small cut of the chorionic villi, which is part of the lining of the uterine wall. Now, this has a slightly greater risk of triggering a miscarriage, but it is still statistically minute. It's kind of like the chance of like a storm making you have a miscarriage. Like, yes, it's technically possible, but other things most likely have been going wrong before this. Now, we have a lot of non-invasive screening, which which we tend to go and test the mother. It's better to test the mother before pregnancy. Because in theory, we also could sort through her eggs and find one that has healthy genetics and use that. But there's also a chance of false positives because again, we're building probability projections. Newborns also tend to go and be blood tested. This allow it basically certain babies need to eat certain things if they have certain genetic disorders. If they are given the wrong food when they have these genetic disorders, it can lead to death, it can lead to mental illness, it can lead to a lot of different problems. So we screen for a lot of the more common ones once the baby's born, just to figure out what treatments they're going to need. It's better if you can go and figure all this stuff out before you even get pregnant. It's better if you go, in general, go to your doctor. If you're pregnant, take your prenatal vitamins, talk to your doctor frequently, get assessments. It's the best thing you can do. We also have a lot of predictive genetic testing on the market. However, you need to be extremely careful with that. 23andMe is like one of the more famous ones. A lot of those genetic guys have been getting in a huge amount of trouble because they will sell genetic information to insurance companies, which then allows them to they basically will either rescind your offer or offer you a new contract at a much higher premium because you are considered a greater potential expense. So don't take those tests. They will sell your data and it will affect your ability to get healthcare. So these are what we hopefully have covered in this. I would like you to be able to define and use the terms homozygous, heterozygous, dominant, all the ones up there. I want you to understand the process of using a Punnett square. I want you to understand the process of manipulating genes. And I'd like you to develop an opinion on prenatal care, postnatal care, how genetics should factor into patient care in general. If you start problem solving using these skills, they'll cement themselves inside of you. Nothing is retained unless it's used. That's another principle of biology. Alrighty, folks. So I'm going to wrap up there. Remember, we have our exam this weekend. I don't want to go and get any emails on Sunday night saying, oh dear, I didn't do the exam. Can I have an extension? You are hearing about this a full week ahead. You got this. I believe in all of you. If you run into an issue, send me an email. We'll figure it out. You might see some repeat questions between the lab and the lecture. The exams, that's just because I kind of wanted to give you guys a freebie. All right. So have a great day. Bye bye.